On to Chapter 12, Nervous Tissue. This will complete the material for exam number 4, which will include the Chapter 10, Muscular System, the Muscular Tissues, as well as Chapter 11, split in 2 parts, Parts 1 and 2 for the Muscular System. Chapter 12 will finish all the material that will be included for exam number 4. So your nervous system, in conjunction with the endocrine system, basically have maintained homeostasis, or at least a balance point for homeostasis throughout the body. The nervous system is considered to be more the fast-acting system, while the endocrine is more the slow-acting system. But the nervous system pretty much is responsible for all of our learned motions, all our behaviors, all of our memories, personality, balance techniques, so on and so forth. And pretty much neurologists and neuropathologists are going to deal with the function of the nervous system and the disorders of the nervous system, respectively. So overall, we're looking at 2 different partitions of the nervous system. You've got the central nervous system, which is composed of just the brain and the spinal cord. And the peripheral nervous system is going to be everything outside of those 2 structures. So the peripheral nervous system will include all 12 of the cranial nerves, 31 spinal nerves, special enteric plexes found in the small intestine, which is kind of nicknamed as the little brain or the gut brain, though it's not the best way to describe it that way. But also all the sensory receptors we find in the skin, as well as other sensory receptors we find throughout the body. So sensory receptors you may be familiar with. These are your deep touch, your vibration sense, heat, cold, pain. Other receptors we find within the body too. If you recall the tendon organs we had talked about in the last chapter, the muscle spindles, also the proprioceptors we have in our joints. These are all position sensors that help us out as well. These are all considered to be part of the peripheral nervous system. So the main structures again, so you have 12 pair of cranial nerves. These will branch off different portions of the cerebrum, midbrain, the brain stem. You got 31 pairs of spinal nerves that branches off the spine. So you have cervical nerves, thoracic, lumbar, sacral, and 1 coccygeal. Basically you have ganglia, which are going to be cell bodies of the actual neurons themselves, going to be found outside of the brain and spinal cord, and I'll show you special ganglia in particular, just off the spinal cord, both from the posterior and also the anterior areas. You have special plexes that help regulate the digestive system, and there are also special plexes also found in the cervical area, the cervicobrachial area, lumbar and lumbosacral areas as well. We'll get to that in another chapter or 2. Now, they're also partitioned as far as what they actually do. So we have sensory, known as afferent, AFF, receptors. These are neurons or specialized cells that monitor changes in both the internal and external environment. So this could be your special senses, it could be the body senses as well. You have interneurons, or what we call association neurons. These are usually in what we call integrative centers, which are usually going to be in the central nervous system, either in the spinal cord or the brain, sometimes both. And you have output, or motor, or the efferent division of the central nervous system to the peripheral nervous system, efferent, EFF, to effectors. So muscles, whether it be cardiac, smooth, or skeletal, and also glands as well. Here's the partition again. So what happens is we get some kind of stimulus from the inside or outside environment. This will trigger the sensory division, partition from either the somatic or the special senses. So we receive sensory input. It gets processed in the central nervous system, sometimes as quickly as through the spinal cord, and then back out to the peripheral nervous system on motor output. Sometimes this information goes up the brain as well for it to take in information and act upon it as well. We'll see a little later on that you, from an evolutionary standpoint, you do want a quick response at the spinal cord level, because it simply takes too much time to actually transmit information up to the brain and back. This is more like a survival mechanism that our bodies have developed over time. So anyway, going back to the motor division, you've got voluntary control through the somatic nervous system. This controls skeletal muscle. And then behind the scenes, involuntary, you're going to have motor input into the autonomic nervous system. Now, out of those 3 divisions there, you see sympathetic, parasympathetic, and enteric nervous system. The enteric nervous system, that'll be covered in a different chapter, gastrointestinal, usually more so in the HAP 2, which controls smooth muscle and the glands of the GI tract. We will focus on the sympathetic and parasympathetic systems. They generally will control the smooth muscle, cardiac muscle, and glands. These 2 work involuntarily. They work behind the scenes, and for the most part, they work in opposing actions for the most part. Although parasympathetic has a little more control over certain things than the sympathetic nervous system, but very simply, the sympathetic is when you're very active, fight or flight. Parasympathetic is what we used to learn back in school, either feed and breed or rest and digest. So again, central nervous system, brain and spinal cord. Peripheral nervous system is going to be the cranial and spinal nerves. These can be a combination of sensory and motor, sometimes dedicated sensory or motor, but oftentimes they're going to at least some kind of integration of both. It's going to connect the central nervous system to all your muscles, the glands, and some sensory receptors. So again, the motor or efferent divisions of the peripheral nervous system. The somatic nervous system is control all the skeletal muscles. This is voluntary. All the involuntary is going to be controlled through the autonomic nervous system. So again, differentiation into the smooth cardiac muscle as well as the glands. So your sympathetic nervous system will generally focus most of its attention, resources, blood flow to fight or flight systems. So this could be dilation of the eyes so we can see appropriately. It's going to be to our balance center. It's going to be to our skeletal muscles to get us either out of danger or prepared to fight ourselves out of way of danger. Now on the flip side, on the opposing action, you have the parasympathetic nervous system. These are known to spend most of resources into the feed and breed or the rest and digest areas of the body. So this can include genitourinary. It can work on the gastrointestinal, the endocrine systems. These are all involuntary, and then they basically will help kind of repair our systems, help to utilize nutrition, so on and so forth. And of course, the enteric system along the GI tract, this independent system kind of works on its own, but does get some information or is regulated by the autonomic nervous system. But for the most part, sympathetic, parasympathetic systems will generally oppose each other. Now, different functions of the nervous system. Again, this is a one-way pathway. So what ends up happening is our afferent neurons will detect stimulations and will be utilized as a sensory function, senses changes and from the internal and external environment, and will send it to the central nervous system. It'll be integrated by association or interneurons, where basically it'll kind of package the information and disperse it to the appropriate areas of the central nervous system. Then once we act upon that information, that's where the motor function comes into play. That's the response to the stimulation. We have motor or efferent neurons to serve the function, again, going to muscles or glands. So your basic component of the nervous system, of course, is a neuron. This is your functional unit of the nervous system. So these have the capacity to produce action potentials. They also have the electrical excitability by stimulus or change. And aside from muscles, from muscle cells themselves too, which can carry an action potential, the neurons are ones that will generate an action potential, at least start it off. The neuron cell body can be named by a prokaryon or a soma. They are usually going to have a very prominent nucleus and even more prominent nucleus because it's going to be constantly churning out new proteins, new neurotransmitters, so on and so forth. Now, if you compare these to muscle cells have a very large smooth endoplasmic reticulum, known as the sarcoplasmic reticulum, but the neurons themselves have a lot of rough endoplasmic reticulums, a lot of free ribosomes for consistent protein synthesis. They're constantly churning out new neurofibrils for the shape of the neuron, as well as increasing more and more neurotransmitters, and also increasing all the different enzymes to break down these neurotransmitters as well, known as the Nissl bodies, or the free-floating ribosomes. The neurofibrils give the cell the shape and support, similar to myofibrils, except they're not contractile tissue that gives you the framework. They have microtubules that will move substances back and forth throughout the cell, whether it be waste products or whether it be nutrients. Because there is a heavy amount of protein synthesis and also protein degradation, a lot of times you'll get a buildup of lipofuscin pigment, which is all the lysosomes that are found within neurons as well, as they're constantly recycling neurotransmitters, proteins, and so on and so forth. And of course, neurons are also very distinctive because they have dendrites, little branch-like projections that will come off of the soma, or sometimes off of a single branch. And in certain cases, they actually may be fused with the actual axons as well. Okay. Most of you should recognize this from the cell chapter, which is chapter 3. A good portion of you had done the neuron as your cell of choice during the Plato lab we done way back. So this is what's known as a multipolar neuron. Multipolar meaning that it has numerous dendrites that come off the cell body. You can see the dendritic spines. These will accept action potential or synapse with either other axons to help carry a nerve impulse. You can see neurofibrils making up the framework of the cell body. You've got the nucleus and nucleus found therein. If you look at a little cone-like projection just to the left of the nucleus, known as the axon hillock, that's known as the trigger zone, where the initial segment where there's going to be a generation of a nerve impulse, that's where it's going to originate. It's going to shoot one way, down through the axon, down towards what we call the axon terminal at the end there, to the synaptic end bulb. Now, synaptic end bulbs can either synapse either with a muscle or will synapse with another neuron. Notice too, these extra cells here called myelin cells or Schwann cells, these will myelinate or insulate this particular axon along this particular neuron. This will help generate a faster action potential down the entire length of the axon. We have these particular myelin sheaths. They leave some exposed portions of the axon called nodes of Ranvier. This is where most of the voltage-gated channels will be exposed to the extracellular fluid. This little exposure here helps accelerate the jump or the leap of the actual nerve cell transmission from one end to the next end of the axon. So axonal transport, we see this in two types of flow. We see naturally in cell bodies here all the protein synthesis, neurotransmitters, repair proteins, structure proteins, what have you. The axonal transport system works in two ways. You have slow axonal flow, where that's where you have movement in just one direction only, so it's away from the cell body. It's roughly 1 to 5 millimeters per day. This is when you're doing repair of the actual axon. Yeah, fast axonal flow. This is where most of the proteins are going to be traveling down through the organelles and all down the surface of the microtubules. Very fast, about 400 millimeters a day. But this can transport in either direction. So it's basically used for recycling. So you're bringing out nutrients to the periphery, and then you're bringing the waste products back towards the cell body. So as far as structural classification neurons, we see three different types. We see multipolar, which is what we just saw a slide or two ago. This is where you have multiple dendrites or multiple branches coming directly off of the soma. They have one single axon. You find these in the central nervous system, and especially as motor neurons in particular. Or you can have what's called a bipolar neuron, where there's one dedicated dendrite or one stalk, which may actually branch out on its own, but there's one stalk off of the soma, and then one axon usually are in direct opposition to the dendrite as well. You see this in the retina of the eye. You see this in also special sensory organs as well, which we'll get to later on. And occasionally, though, you'll get what's called a unipolar neuron, where basically the cell body is just offset, and there's one stalk, which basically one end of the stalk is going to be dendritic, and the other is going to be more of an axon. You find these as sensory nerves, in particular the sensory nerves out into the periphery, whether it be your touch sensors, whether it be for smell, taste, where the cell bodies are going to be closer to the spinal cord and the cranial nerves. And usually these, where most of these cell bodies will congregate, will be in a ganglion, which will show a little swelling, especially around the spinal cord, or especially around some of the more of the proximal end of the cranial nerves. So here's what the three neurons will look like. If you notice, each one has a little space there. It simply means that the length of the axon can vary very much so. In fact, actually, the longest cells in the body are to be neurons. So basically, when I'll show you what like a sciatic nerve will look like, or the axillary nerve, or the musculaneous nerve, these nerves in particular, they're basically made up of axons that travel from the spinal cord out to the periphery down the extremities, or sometimes down the trunk. So these axons can oftentimes be very, very long. But as you can see on the left side, letter A, the multipolar neuron, the way you can pick these out is as multiple stalks of dendrites off different areas of the soma. There's a trigger zone, the axon. Of course, all three of them are actually myelinated with the axon terminal. Of course, the axon can have multiple synaptic end bulbs to either synapse with a number of different neurons or possibly muscles too. Letter B, the bipolar neuron, you can see one dedicated dendrite that comes right off the soma, but that will branch out on its own too. You see the same trigger zone and, of course, the same length of the axon and the different axon terminals there as well. Now, letter C, unipolar, this is where you're going to see the cell body offset. You see one stalk come off of the cell body, but it's a fusion of the dendrite as well as the axons, or the axon, excuse me. So it's actually one solid piece. You see these in almost all, it's almost always going to be a sensory neuron. You won't see this as a motor neuron. So again, functional classification, so we have three types, and this should be set up a little differently. It should be sensory neurons first, just by virtue of how the impulses are generated. They are afferent. They send the action potential excuse me, to the central nervous system. Then the interneurons come into play. This is going to be the bridge between the sensory and the motor, otherwise known as association neurons. These can be either inhibitory, meaning they can actually shut off a certain nerve impulses, or they can be excitatory. They can generate other nerve impulses. But based on the interneurons, they will send information down or send an action impulse down the motor neurons, or an efferent away from the central nervous system to the effectors. Now we'll see this kind of thing again in a couple different slides here, but very simply again, you look at a sensory receptor in the peripheral nervous system. This is a unipolar sensory neuron. Usually there's some kind of conformational change with the actual sensory receptor. In this case, let's say if it was a Pacini corpuscle for a vibration sense, the vibration will generate the actual potential that'll travel from the axon towards the central nervous system, so it'll pass by the cell body. It'll synapse with an interneuron, sometimes either in the spinal cord or in the brain. The interneuron will send another impulse down towards a motor neuron, usually multipolar, and then send it out to the effectors, whether it be, again, any one of the muscles found in the body or any of the glands. These are two specialized neurons. All someone would know is just what they're called. The Purkinje cell, we can find this in the cerebellum, which is our major balance center. This is named after the histologist who first came across this. The right cells, called a pyramidal cell, you find these in the cerebral cortex. Very similar, called pyramidal cells because the cell body looks very similar to a pyramid structure. Other than that, just be aware that these two are both found in the central nervous system. So let's talk about some of the support level cells. If you guys remember when you guys looked at these slides way back when, there's a huge neuron with a lot of small cells found therein. These are neuroglia. So it's kind of to tell on the slide which ones are which. But very simply, you have different cells with different functions. You have astrocytes, named because they look like a star. They can be found either as in the gray matter as protoplasmic or be fibers in the white matter. These are your support level cells. They basically help maintain the blood-brain barrier. They basically aid in any kind of development for embryological development. They help maintain the chemical environment and also aid in the formation of synapses or trying to form memories. Now in the central nervous system, you have oligodendrocytes, which are specialized cells that will provide the myelin sheath or insulation around certain types of neurons. Now these are basically akin to the Schwann cells, but Schwann cells are only found in the peripheral nervous system. The oligodendrocytes are also different in the sense where they will myelinate different nerve cells at the same time. So they may branch off into 2, 3, 4, possibly even more axons of nerve cells, where a myelin, one Schwann cell will wrap itself around one particular axon. You have microglia, which are basically phagocytes, which will take care of the kind of foreign material. And last, you have the epidermal cells. These are specialized either cuboidal or columnar epithelial cells. You have both microvilli and cilia. These help produce your cerebral spinal fluid. So these are constantly recycling the CSF. You don't find these in the peripheral nervous system because your cerebral spinal fluid will only encase or bathe the brain and the spinal cord, in fact, in a very continuous loop, which I'll show you later on. CSF is also very delicate in the sense where you don't want too much, but not too little, because that can cause all kinds of disruption with the brain and spinal cord. Here's a quick little shot here. They're neuroglia from the central nervous system. So you see up top there the cells of the pia mater. The pia mater is going to be the innermost covering of the brain and spinal cord. So you can see here the neurons in yellow. You can see the microglial cells interspersed throughout. These are your phagocytic cells. You've got astrocytes, which are going to be found both fibrous and also protoplasmic. You can find these along the white and also the gray matter. And finally, you'll see in pink, you'll see the oligodendrocytes. You see two of them there are going to be branching off into different myelin sheaths along the individual axons of the neurons. And finally, you're going to see the epidermal cells down on the bottom of the diagram. You can see the microvilli and the cilia. Again, these will help to produce the CSF and help recycle the CSF, and also helps as a partition off the brain and blood barrier. So neuroglia of the peripheral nervous system, we generally have two types. You just have the Schwann cells, which will encircle the peripheral nervous system axons of myelin. And you also have satellite cells. These are basically do the functions of all the other cells we mentioned before in the central nervous system. So they also will help support the framework, work as phagocytic properties, and also aid in the chemical environment. But one thing I want to point out, though, is the Schwann cells can either myelinate the axon and form the node ramifier, or what they may actually do is actually encircle the unmyelinated axons. So they can still enshrine the unmyelinated axons without providing myelin. So these will remain as what we call gray matter or as unmyelinated axons. We'll see what the reasoning for the myelin sheath, why you want it there in certain actions and certain elements, and why not in others, a little later on in the chapter. So myelinated and unmyelinated axons. So your Schwann cells will myelinate the PNS during field development. This is very important for axonal growth. Also helps increase the speed of nerve conduction. Schwann cells of the cytoplasmic nucleus will basically have an outer lemma called the outermost layer named neurolemma. The innermost layer is the myelin sheath. So the myelin sheath is going to come in contact, direct contact, with the axon itself. So this also aids in regeneration along that particular tract. And if there is any regeneration left in a neuron, the neurolemma has to be present to create what's called a regeneration tube, so the axon can rebuild itself along that tract. The nodes of Ranvier are going to be the exposed nodes in between the Schwann cells. It's necessary to have these there to allow some kind of neurotransmission of the action potential, but literally leaps from node to node, so it's actually a very fast transmission versus a axon that does not have any kind of installation. And of course, neurolemma is not found in CNS because Schwann cells are not found in the central nervous system. So, the answer to the first question, is it possible for nerves to regenerate? And if it is possible, where would you expect regeneration to occur, either central nervous system or peripheral? Why or why not? And in particular, what part of the neuron can grow back, if at all? So I kind of just answered that question here. So we see better regeneration rates in the peripheral nervous system because when you have the Schwann cells there, they will lay the groundwork of the myelin sheath to allow the axon itself to start to grow. Now it's a slow process, roughly about 1.5 to like 5 millimeters per day. It's not a lot, but it does have them regrow. So some neurosurgeons in particular can put down some myelin sheaths or lay down a track to actually grow back portions of the neuron. Now again, it's a very lengthy process. Trying to retrain a person that has had any kind of nerve damage can take a long amount of rehab, but it has to be the axon for it to grow back. You can grow back dendrites up to a point as well, but the biggest problem is this: if the nerve cell body is dead or necrosed, then there's no way you're going to regenerate the axon. So as you can imagine, as we get older, then any kind of regeneration, if it happens at all, is hampered as we age. If you're able to have any kind of injury or sustain injury when you're younger, before you're fully formed, then you have a better chance from a plasticity standpoint of regrowing some particular nerve tracts, especially up and down the spinal cord, or actually nerves themselves through the peripheral nervous system. But once you're fully formed, it's only the peripheral nervous system, for the most part, that can regenerate. There's very few spots in the brain, which I'll talk about in a few minutes, that can regenerate, but it's a totally different mechanism. Here's a nice little shot of both a myelinated and unmyelinated axons. Pay attention to letter A in particular. You'll probably see this again, probably on your next exam. Notice the node of Ranvier, which is going to be the exposed area of the axon, while the rest of the nucleus is just offset. The Schwann cell, you see the Schwann or the myelin sheath will wrap itself in numerous layers around the actual axon. And then letter B, you can see one particular Schwann cell is going to somewhat encapsulate multiple axons, but no myelination is there. There's no myelin wrapping around the individual stalks of the axon. And of course, you can see transverse electron microscopes of both the myelinated to the left, unmyelinated to the right. Gives a nice little picture, of course color-coded, to give an idea of what it would look like in situ. So we talk about collections of nervous tissue. As I mentioned before, it's going to be the axons that are going to create nerves and tracts. So first, let's talk about the nerve cell bodies. So we have ganglia, which are going to be clusters of nerve cell bodies found in the peripheral nervous system. There's a special ganglion found just below the spinal cord called the dorsal root ganglion, where all the nerve cell bodies will cluster there, and then from there, it'll create the nerve that travels down the trunk, down the upper extremity, down the lower extremity. So your sciatic nerve is actually a large band of axon bundles with all of the cell bodies, at least from the sensory standpoint, found in the dorsal root ganglion. But you also find different ganglion as well in the autonomic nervous system, up and down the sympathetic trunk. There's also some for the parasympathetic as well. We'll get into that when we get into the autonomic nervous system. Now, if we're dealing with nerve cell bodies found within the central nervous system, these are called nuclei. We found these mostly in the brain. And we have different tracts of axons that travel both up and down the spinal cord. In fact, there's different sections of the spinal cord. If we took a cross section, you can identify where the tracts are. So generally, if someone has had any kind of nerve injury or any kind of spinal cord injury, we can generally get an idea of what kind of symptoms they may be having based on what tracts are being affected. And of course, we're going to have interspersed within all the nerve tissue of the spinal cord and also the brain, you're going to have gray and white matter. So your gray matter is going to be unmyelinated, and your white matter is going to be myelinated neurons. Here's a nice little shot first of the transverse section of the spinal cord. So the gray matter forms almost like an H pattern. So if you're looking at the spinal cord here, the right side is on the left, and the left side is on the right. The posterior portion is going to be upward on the diagram here. The anterior portion, a little bit of a sulcus there, is going to be the anterior portion. We'll get more into that anatomy when we get into the brain and spinal cord. But you can see interspersement of the white matter around the gray matter there. And of course, it's a little more interspersed in the brain. There's going to be a coronal or frontal section of the brain. Okay, so let's talk about electrical signals found in neurons. So basically, the neurons are excitable because there's a voltage difference across the membrane. So we see two types of electrical signals. We see action potentials that can travel the entire length of an axon. We have graded potentials, which are very localized, which very simply change or can only really disperse through a short portion of the neural lemma. So basically what ends up happening is if there's any kind of membrane change or electrical difference, we have different ion channels that are open, that can actually generate a graded potential. So this can be done by either electrical gradients, or it could be by pressure, vibration, or any kind of binding by different types of ligand-gated channels. So for example, like smell, taste, certain types of proteins can actually link up and then basically open up some ion channels and therefore cause some kind of graded potential. So in this case, we're looking at the sensory receptors in the finger as the person holds the pen. So judging on how strongly they're holding onto it, or let's say what kind of action you're doing or what you're writing or drawing, that kind of sense or that sensory input coming in from the pushing onto the pen will trigger the sensory receptors in the fingers. They will charge a graded potential that will travel up towards the sensory neuron, and then that'll integrate with the association or the interneurons found in both the spinal cord and also in the brain. So those are association or interneurons. They will synapse with the cerebral cortex, and then that will actually further synapse with the motor or the efferent neurons. That'll send information out to the muscles to generate what kind of action you're trying to do based on the stimulus. So in this case, it could be drawing a certain word, or it should be scribbling something out which you fear you made a mistake. But as you can see, it's a one-way system. So it goes from the sensory area, you trigger off the sensory neuron to interneurons in the central nervous system, and it transmits an efferent pulse or efferent impulse down the motor neurons, in this case, to the effector, meaning the skeletal muscles. So let's talk about different kinds of ion channels that we find that help maintain electrochemical gradient. So first you have leakage channels. These will alternate or open and close randomly. We have more potassium channels than we do sodium. The permeability of potassium is higher. This allows you to maintain a negative 70 millivolt resting membrane potential in nervous tissue. This is known as being polarized. Other than that, you have ligand-gated channels, open close in response to different types of stimulus. So it could be a neurotransmitter, could be a glucose molecule with taste, it could be different odorants when we smell, but something that basically will open up those ligand-gated channels and allow either sodium to cross or potassium to cross over the plasma membrane. You also have mechanically gated channels, which respond to mechanical vibration or pressure. We find these in ear that deal with sound. We'll talk about those later on. You can also have voltage channels that respond to any kind of change in membrane potential. So this works very similar to the neuromuscular junction we saw back in chapter 10, where acetylcholine will basically be released in the synaptic cleft. It will bind with ligand-gated channels and allow sodium to rush in, therefore generate another action potential. So here are the ion channels found in the plasma membrane. So very simply, you're looking at a leakage channel for potassium on the first slide there. Next, you have the ligand-gated channels. So in this case, acetylcholine will bind with the sodium channels and allow sodium to rush in and for potassium to rush out. You can have mechanical gated channels. So basically, any kind of change in the conformation of the plasma membrane can open up these channels here. So it could be pressure from deep touch, it could be vibration, it could be sound, it could be movement based in the ear, which we'll talk about a little later on. But occasionally you might also have a voltage-gated channel where an action potential will basically open up the voltage-gated channel here, in this case potassium, allowing potassium to rush out of the cell, and that will actually depolarize the actual neuron. So again, your resting membrane potential is otherwise known as a polarized cell. These are going to be at a negative 70 millivolt grading, and so basically your negative ions will align itself alongside the inside of the membrane, and positive will align itself outside. This is because of the concentration of the ions between the extracellular fluid and the cytosol. So the extracellular fluid has a lot of sodium, a lot of chloride looking to get into the cell. The cytosol is full of potassium, phosphates, amino acids, and the potassium is always fighting to get out of the cell. So besides the leakage channels, you also have the sodium-potassium pumps that are constantly trying to pump in the potassium, pump out the sodium, try and maintain that polarized state. So again, you can see the leakage channels both for potassium and sodium. You can see the sodium-potassium pump on the far right. So again, this is constantly using ATP to generate the action potential there. To generate the right kind of balance and going against the concentration gradients for both sodium and for potassium. So we have a graded potential. These are just small deviations from the resting potential of negative 70 millivolts. So it goes in one of two directions. It can either become more negative, which means it's hyperpolarized, which means you need a greater stimulus to get an action potential to occur, or it becomes depolarized, where it becomes more positive, so you're getting closer to what we call threshold, which makes the nerve a little more excitable and a little easier to stimulate. So these can vary quite a bit depending on the strength of the stimulus, and this could be either strength of, let's say, multiple stimuli coming at the same time. Let's say if you have a really heavy weight that you're trying to hold, so you have multiple stimulus patterns coming from different parts, let's say your hand, or the strength of repetition of a particular stimulus, where if it keeps getting stronger and stronger, eventually the graded potential will actually combine to actually fire off a threshold response. So when we say localized, these particular changes in the action potential will spread across the plasma membrane, but what winds up happening is as it passes by a short distance, we get what's called decremental conduction, where it kind of fades away a bit. So graded potential is somewhat less than an action potential. It'll kind of die out if there's a lot of surface area for it to travel over, unless it has a lot of other help with other graded potentials along the same nerve or nerve cluster. So here we see, at least from a graph standpoint, both the hyperpolarization and depolarization potentials. So again, if it becomes hyperpolarized, that means it's going to be tougher for us to maintain a threshold response. So in this case, it's going below negative 70 millivolts. So in other words, there's more of a gradient to travel across to try and actually fire off the neuron. Or it goes in the opposite direction. We have depolarization of the graded potential, where it gets up to just under negative 55 millivolts. Negative 55 millivolts is what to remember to actually achieve an action potential. So this is what we call subthreshold. So it's enough to kind of get it, give it a little bit of help, but not quite to generate a full action potential. So we see different graded potential response to different types of mechanically gated and ligand gated channels. So for example, if we have a mechanically gated channel, we apply a little bit of pressure or some kind of conformational change to the cell body. It will open up these channels and it will make things a little bit easier to depolarize a particular nerve. Occasionally, though, you may also have acetylcholine that may be helping out. These will open up sodium gated channels again to again make the cell a little more excitable to allow it to fire more easily. Or occasionally we'll get inhibitory type of neurotransmitter, like in this case glycine, which actually will open up certain channels, like in this case chlorine, to actually make it more negative inside the cell and make it more hyperpolarized, which means we need a very strong stimulus to fire up this particular nerve. So let's start with different types of stimulus strength and graded potentials. So in this one here, so again, the magic number that we're trying to achieve is negative 55 millivolts. These are all what we call subthreshold stimuli, although each one gets a little stronger as time goes on. Each one of them is going to generate a little bit of graded potential, meaning there's going to be some kind of change in the resting membrane potential of the nerve, but not quite enough to cause the actual firing of the action potential. Remember, we have to hit negative 55 millivolts to activate the action potential. So right now, these three are simply graded potentials because there's still some kind of change to the electrochemical gradient. Now, occasionally, through what we call summation, and there's two types. There's going to be spatial and there's going to be temporal, which we'll get into at the end of this chapter here. So basically, if you add two graded potentials together, the amplitude itself will increase, especially if it's sustained. So if you notice these two here, stimulus 1 and 2, when they're built upon one another, you're still going to be sub-threshold, but it's stronger than each individual one by themselves. Now, action potential to impulse is an all or nothing type of principle. We're going to see basically a polarized membrane is going to hit depolarization at negative 55 millivolts, and then at some point the nerve is going to fight to get itself back into a repolarized state and back to a polarized end position. During this particular time, it's what we call an all-in principle, where the stimulus reaches threshold and the sequence has to follow in a certain sequence of events to cause the action potential. And then basically what winds up happening is you have one type of period called a refractory period, where it absolutely will not generate another action potential until it finishes getting itself back into close to a polarized position. But very simply, what winds up happening is you have sodium and potassium channels that are going to open. One moves a little faster than the other, which we'll look at in a couple of seconds here. But it is that interaction between the sodium and potassium that allows it to get through the different stages of the action potential. So let's look at different stages here. So we start off first in the stage where the cell is relatively inert. It's a negative 70 millivolts. You get a stimulus, and once you hit that negative 55 millivolts here, the cell all of a sudden depolarizes. So what winds up happening is your voltage gauge channels, the sodium channels will open up, and then as they start to close, that's when the voltage gauge channels will open up there too. That's what we call the absolute refractory period. There cannot be another generated action potential until we get past this particular period. Then once we get to the relative refractory period, this when the voltage channels of the potassium are still open, but the sodium channels have closed or back at rest. And if you notice underneath the after hyperpolarizing phase, notice that it dips down a little bit below the resting membrane potential. So we say that the nerve is hyperpolarized at that point, but if you have a strong enough stimulus, you can still fire off another action potential once it gets to the relative refractory period. But absolutely under no circumstance for the absolute refractory period will get the nerve to fire off a second action potential until you get past that particular stage. So let's look at different types of stimulus that we can achieve here. So one is subthreshold stimulus. As you see here, it's not hitting the threshold point at negative 55 millivolts. It's a great potential, nothing much happens. However, if you get enough to actually cross threshold, you have threshold stimulus, which is just a short stimulus there. So pretty much once the cell returns back to the polarized state, it just stays there until another stimulus hits. Now in blue here, we see superthreshold stimulus. This is where the stimulus stays there. So in other words, the nerve will keep firing off back and forth, back and forth. It'll keep going through that particular stage until the stimulus is taken away. But if you notice here, especially in between each of the action potentials, the spikes you see there, 1, 2, 3, there's still going to be one point where it's going to drop down below threshold. That's going to be the absolute refractory period until you get back closer to the negative 70 millivolts for it to fire off again. So let's start with different stages here. So you notice in the diagrams here, there'll be a different portion of the graph just underneath them. So first you have the resting state, where basically you have the sodium channels alternately closing and opening. So you have both an activation gate and also inactivation gates are open and closed. So right now it's still at negative 70 millivolts. Once that depolarizes, the sodium channels will open up. So both gates of the sodium channels will open up. Sodium rushes into the cell. And then once the nerve cell wants to try and get itself out of the depolarized state and go back into repolarization, this is when the sodium channels will close. Your potassium channels will open to try and get some more negativity on the inside portion of the plasma membrane for the cell. And at this point, once the sodium channels are back to a resting position, that's when you're back in the repolarization phase. You actually can generate another action potential at this point. But so once you get past the repolarization phase, once you get back to the negative 70 millivolts or very close to it, that's when the potassium channels will close again. So again, depolarizing phase, either chemical or mechanical stimulus causes the action potential to reach negative 55 millivolts. So the voltage gated channels of the sodiums will open, sodium rushes in. In the resting membrane, interaction gate, the sodium channel is open and the activation gate is closed. When the threshold reached, both open and the sodium will rush into the cell. Now these close within a couple 10,000ths of a second. Only about 20,000 sodium molecules actually enter the cell, but that's enough to cause a conformational change, and the potential rises up, sometimes up to positive 30 millivolts. This is a positive feedback process, so it ramps up to a certain point, and once stimulus is done, then it drops back down again. So repolarization phase, once you hit the negative 55 millivolts, that's when the voltage gated channels of potassium open up. So in other words, again, we're trying to fight to get more negative side on the plasma membrane within the cytosol versus the outside. So again, the potassium channels, as they open up very slowly, that's when the sodium channels will start to open as well. Once the potassium channels finally open, sodium channels close down. So now the flow of the sodium stops, but the potassium flows back out. It returns the membrane potential back to negative 70 millivolts. But a lot of times what happens is that overshoots the mark. So when you have too much potassium that leaves the cell, it reaches a negative 90 millivolt membrane potential, and then you're in the after hyperpolarizing phase. But again, at this point, once sodium channels are closed, you can achieve another action potential. Once the potassium channels were closed, the membrane potential will eventually return back to rest potential as long as there is no more stimulus. So again, the refractory period is a period when the neuron cannot generate another action potential. The absolute refractory period, again, doesn't matter what stimulus you have, it has to go through this stage of allowing the sodium channels to inactivate and return to the resting state. Now, some types of fibers or nerve fibers found in the central nervous system have a slower refractory period than others. The fastest refractory period is going to be in the large fibers. These are what we'll call your type A fibers. It's a 0.4 millisecond, and it can actually have up to 1,000 impulses per second, as long as there's still a stimulus being generated. Once you get to the relative refractory period, this is when the sodium channels are closed, but the potassium channels are still open. You can have, again, if it's strong enough stimulus, you can generate another action potential. Let's talk about different types of propagation or conduction down the nerve fibers. So if you remember before, I talked about unmyelinated versus myelinated fibers. So when you have a unmyelinated fiber, you have what's called a continuous conduction, which means step by step, all along the surface of the cell membrane, each gated channel in succession will open up. Whereas with the saltwater conduction, depolarization only happens at the nodes of Ranvier, the exposed areas between the Schwann cells, where there's going to be a very high density of voltage-gated ion channels. So this basically carries the axon flow very quickly, and it's almost as if the generation of action potential will leap from node to node to node. So it basically acts like insulated wiring. So this allows two things. One, a faster transmission speed, and number two, it doesn't allow the signal, so to speak, to jump from one nerve to the other. In some cases you want that jump, in some cases you don't. So let's look at the nerve cell, the neuron on the left. So this one is going to be continuous conduction, where as it follows down each and every segment of the axon and goes through each individual sodium channel. You look at letter B, the saltwater conduction. This is going to be a myelinated neuron. So you see it's going to jump from one node to node to node where the exposed sodium channels will be there. And this also increases too, like the larger the fiber, the more surface area, which means the more sodium channels will open. That can generate a faster action potential down the axon. So again, other factors that are going to affect the speed of the propagation is one, the amount of myelination. So the more insulated it is, the faster the transmission. The larger the nerve means the greater surface area, which means going to be more sodium channels that can be open to generate the potential. Now this is going to be an outside factor, an environmental factor. Faster or higher temps also will generate a faster impulse. This comes as no surprise to most of you, especially when you start thinking about when you're cold. If you ever hurt yourself when you're cold, it takes a little bit longer for the pain response to generate versus say in the summertime. Another thing too is if you look more from the motor aspect, when it's pretty warm outside, it's easier for you to warm up during a certain activity. Versus if it's cold outside, it might take a little bit longer to actually generate heat and actually get the muscles to move a little more thoroughly. Some people do tend to go into spasm or cramp sometimes in the winter months versus summer. Although you do see some summer cramping, but it's usually more so to dehydration, not necessarily the temperature. So again, if we look at the speed of the impulse of the action potential, it's not related to the amount of strength in the stimulus. It has to do with how the actual structure of the axons are formed. So typically type A fibers are largest. These are going to be myelated, somatic, sensory, and motor. Usually it's sensory muscle, to skeletal muscle. Has a relatively small refractory period. Your B fibers are going to be the intermediates. These are also myelinated. These usually go to the viscera. They're going to be autonomic neurons as well. Your C fibers are going to be the smallest. They're going to be unmyelinated. Generally, these are for sensory and autonomic motor. These are also very important because they're very important for pain. So very important in any kind of line of work we're dealing with any kind of patient that is dealing with recovery from pain. There are certain techniques that we use that will utilize the A fibers to basically outpace the C fibers to basically block the pain signal. Best analogy I can give you is if you scratch an itch. You know the itch sensation is not pain per se. You're putting a mechanoreceptive input to basically override the actual itch response. That's why a lot of times we scratch an itch. So two types of synapses that we see. The first one is going to be electrical. If you remember these are going to be in the gap connections through connexons. Literally, you have direct communication between one cell and to the next. It's a very fast two-way transmission, and you can synchronize a bunch of different neurons at the same time. But the majority of synapses that we see are going to be chemical. These are one-way information transfers. It goes from dendrite to axon. It goes from presynaptic neuron to a postsynaptic neuron. So they can connect either from axon to dendrite, so axodendritic, or axosomatic, which is axon to cell body, or axonic, which would be from axon to axon. So this chart should have been pushed back a little bit, but this gives you a quick little breakdown of the difference between a gradient potential versus action potential. So this is a nice little study guide for you guys to use. Okay, so here we see a postsynaptic neuron that's going to be synapsing either from axon to axonic. You can see an axon directly connected right to the axon hillock of the neuron shown. You have axodendritic, which is going to be from dendrite to axon, or sometimes axosomatic, where it will synapse directly with the nerve cell body. So again, electrical synapses, basically action potential will travel across the plasma membrane via gap junctions. They'll pass the ions from membrane to membrane through the connexons. That's fast communication, and also is good for synchronization from then a chemical synapse. So this should be relatively easy for you. So if you know anything about cardiovascular system, which should give you a hint, where would you expect to find electrical synapses? Most of your electrical synapses are going to be found in the heart, the cardiac muscle. You can find these sometimes in smooth muscle as well, but for the answer to this question, we'll focus mostly on the heart. So basically, the heart connections from the neuromuscular junction or between the neurons for the heart are connected for a faster coordinated response through electrical synapses. Strangely enough, the chemical synapse, which I'll talk about in a little bit, because of the synaptic cleft, the neurotransmitters literally have to travel from one end of the synaptic cleft to the other. That takes up a little bit of time. And unfortunately, even though the short time it may take, it's not fast enough for the brain to act upon any kind of feedback it's getting from the heart and basically let it adjust accordingly. So in the heart, we see a very fast synchronized coordinated response with electrical synapses to allow increased heart rate, increased contraction of the heart rate, and basically allow the body to kind of maintain as best a homeostatic position as possible and respond as quickly as possible there as well. So if you look at the most extremes, you look at people that are athletes or, again, in a very stressful situation, the body responds by activating the heart to either increase the blood pressure or increasing the heart rate to get more blood to the areas that need it. And vice versa too, from a relaxed position, this helps slow down the heart rate so you're not burning resources too quickly and allows you to rest and digest. So chemical synapses we found in the majority of the central and peripheral nervous system, and also the synapses we find at the neuromuscular junction. So with these, you have an action potential that reaches the end bulb, and basically the voltage gated channels of the calcium are going to flow open. This will trigger the release of a neurotransmitter. Now, neurotransmitters can be acetylcholine, very similar to neuromuscular junctions, but it can be other neurotransmitters there as well. So they will cross the synaptic cleft and bind to the light gain receptors, and that will open up the sodium channels on the postsynaptic neuron to allow the next action potential to occur. But there is a synaptic delay. This is one thing you cannot afford, especially in the heart, and it's 0.5 milliseconds. That's an eternity as far as the nervous system is concerned. The other downside is this is a one-way information transfer. So you can't generate an impulse up the same neuron that it just generated from. So again, remember that one slide we looked at where it went from the sensory to the interneurons back to the motor? It doesn't work the other way around, unlike electrical synapses that can transfer information both ways. Here's a nice little shot of a synapse here. So you've got a nerve impulse traveling down one neuron, the presynaptic neuron. So with this, calcium rushes in. It triggers the synaptic vesicles to release the neurotransmitter. These will bind in a ligand-gated channel, so it'll allow sodium to rush in, and you start the next impulse down into the postsynaptic neuron. Now, this kind of synapse can generate two types of potentials. You can have either an excitatory or inhibitory type of response. So excitatory will help depolarize a particular neuron, It results from opening of the ligand-gated sodium channels. It makes it more easily to reach threshold in the postsynaptic cell. So we call it an excitatory postsynaptic potential called EPSP. But occasionally you'll get a neurotransmitter that will be inhibitory or create an inhibitory postsynaptic potential that will actually hyperpolarize the cell. This will open up different channels, in this case, chloride or potassium channels. So it'll actually allow chloride channels to rush in and potassium channels to rush out. So it makes it less likely for the postsynaptic neuron to reach threshold. This is the basis for certain drugs in particular, either to enhance certain types of synapses or to decrease the activity of certain synapses. So here we see a acetylcholine receptor in letter A. It's called ionotropic. So in other words, the ligand-gated channels will bind directly to the gated channel. In this case, allows sodium to rush in. Look at letter B. This is ionotropic GABA receptor. This is an inhibitory neurotransmitter. So this one will allow the opening of the chloride channels, let that rush into the cell. And the last one you're going to see is a metabotropic. This is where you'll have an acetylcholine receptor that will bind to a regulatory protein, in this case a G protein, which therefore triggers the calcium channel to open up. So a little different than direct binding to the channel proteins. This time you're going to have a metabotropic that's going to attach to a regulatory protein, which will have a conformational change and then change the gated channel in a different part of the plasma membrane. Now, another ways to either increase or decrease the effectiveness of a neurotransmitter, the rate of diffusion, how quickly or how much a concentration gradient there is. If it's a very high concentration, you can remove the neurotransmitter relatively quickly. If it's a low concentration, it's a little bit tougher. You also have different enzymes that will constantly recycle a lot of these neurotransmitters. In this case, acetylcholinesterase will help break down acetylcholine for reuptake. And occasionally you have uptake by either neurons or glial cells. You have neurotransmitters or transporters, or occasionally different types of drugs or other neurotransmitters. In this case, for Prozac, serotonin is what's known as the feel-good hormone. People that tend to be very anxious will take Prozac to basically allow serotonin to kind of hang around the synapse a little while longer, to allow more of an action potential to kind of let things kind of calm down. So let's get back into summation. So summation is the culmination of different graded potentials together. If they are being released at the same time, that's called a spatial summation. If you have the same neuron, but it's firing off more quickly and allowing more neurotransmitter to go through the synaptic cleft, that is known as a temporal summation. So again, summation of neurotransmitters is released from a number of different Elm bulbs at the same time, more location of stimulus onto one neuron. And temporal will be the same neuron, but it's going to be a rapid release neurotransmitter after numerous firings of graded potential in rapid succession to the second or the postsynaptic neuron. So we see three possible responses when we do temporal summation. So occasionally you might get a small EPSP, which generally doesn't cause an action potential. This is a graded potential. So it's any kind of potential that reaches anything from negative 56 millivolts and below. Remember, you need at least negative 5 millivolts to achieve threshold. Or you can achieve threshold where the impulse is generated and membrane potential hits negative 55. Where you have an inhibitory process where basically the membrane becomes hyperpolarized and drops below negative 7 millivolts, which, as you remember, makes it tougher to fire the nerve off into an action potential. So here you see on the left, you see spatial summation. You see two different presynaptic neurons. Will synapse with one postsynaptic neuron. If the two presynaptic neurons can fire off enough neurotransmitter across the cleft, then basically the postsynaptic neuron will fire as received nerve impulse there. So if you see one, or something just one or two fires off, but both of them fire at the same time, then generate the action potential. On the flip side, for temporal summation, if you have a presynaptic neuron that just has a general, very quick firing, then nothing else follows it, then it's not enough to cause the threshold to be reached, great potential. But then you have a rapid firing at the presynaptic neuron, where it's constantly releasing the acetylcholine or another neurotransmitter. That should be enough to fire off the postsynaptic neuron for the action potential. Now, neurotransmitters can be either excitatory or inhibitory, sometimes both, depending on where they're found in the central peripheral nervous system. So, in other words, let's take acetylcholine. Acetylcholine in the peripheral nervous system, for the most part, is very excitatory, but in certain aspects, can be very inhibitory in the central nervous system. So, other neurotransmitters that you just want you to be aware of: acetylcholine, glutamate. We find glutamate in particular, especially around the eyes, helps kind of fine-tune the actual images that we interpret and we get to the occipital lobe. aspartate, gamma-aminobutyric acid, also known as GABA, glycine, norepinephrine, epinephrine. These are your adrenals, your adrenal gland hormones, and dopamine, which is very important for sleep patterns. So neurotransmitter effects can be modified by either the synthesis of how quickly they can be stimulated or inhibited, how well they can be released or blocked or enhanced. The removal of certain neurotransmitters, as you mentioned with Prozac, can either be stimulated or blocked, or sometimes you can actually block the receptor site with other neurotransmitters or possibly other drugs. But generally, anything that helps a neurotransmitter's effects is known as an agonist, and anything that blocks the action of a neurotransmitter is known as an antagonistic type of drug or neurotransmitter. So anything that either will prohibit it from binding with something or prohibiting or actually speeding up the degradation of a neurotransmitter is known as an antagonist. So your small molecule neurotransmitters are going to include acetylcholine. You find this in the majority of the peripheral nervous system neurons and some of the CNS. You find these in all the neuromuscular junctions, but can be inhibitory in other areas. Again, inactivated by acetylcholinesterase. Another amino acids, glutamate in particular. These are released by nearly excitatory neurons in the brain, but can be inhibitory in certain areas as well. GABA is a major inhibitory neurotransmitter found in roughly about a third of all brain synapses. So Valium in particular helps have a calming effect here. So basically it aids GABA in inhibiting other synapses. Now, quick little neurotransmitter question. Now, talking about nitric oxide and carbon monoxide, and what little we know about them already. Could you find these normally in the human body, and do you think they would be a benefit, or could they be a harm? Now, the thing to remember is nitric oxide is not the same as nitrous oxide. Those who go to the dentist that still use nitrous oxide, or very few do, especially those using 19th and 20th century used nitrous oxide before they did any kind of oral surgery. We're not talking about nitrous oxide, that's the one that makes you laugh. We're talking about nitric oxide. So strangely enough, both of these can be found in the body. They're found in very, very small trace amounts. They're not prepackaged, and let's talk about the actual benefits of both of these. So nitric oxide, not to be confused with nitrous, is secreted in the brain, the adrenals, and also in the male penis. These are formed on demand. These are basically used for helping to relax smooth muscle and for vasodilation. However, nitric oxide, thankfully because it's not prepackaged and formed on demand, it is very toxic at high levels. Now, carbon monoxide is very important, also not prepackaged, also formed on demand. It actually helps calm down or decrease excessive neural activity, helps aid in memory and olfaction. It's also good, very good for anti-inflammatory activity there as well. But again, this can also be, as we all know, because we all heard about carbon monoxide, especially starting your car, especially in the garage without opening the door, it can be fatal. But carbon monoxide does play a role, at least with memory in particular, and for anti-inflammatory activity. As far as neurotransmitter, naturally, these are the smallest neurotransmitters you'll find. Okay, neuropeptides are known as long-chain neurotransmitters. There's only a handful of these that I want you to be aware of. In particular, is substance P. This is going to be the pain neurotransmitter. Basically, this fights against your enkephalins and endorphins. So the substance P is what you're going to feel when you first sprain an ankle or break a bone. The enkephalins and endorphins will start to be secreted more so to block that substance P. So usually you'll find, within the first 24 hours of breaking a bone or spraining ligament, a lot of times the pain is not as severe as it was when it first happened. It's because the enkephalins and endorphins, respectively, are going to suppress the release of substance P or block it. So this way, it's a great way to help calm the brain down so it doesn't get overstimulated from the substance P being turned too much. Dynorphins, these also help controlling pain, registered emotions. Hypothalamic releasing inhibitory hormones will talk about more so. We get into the anterior pituitary. Angiotensin is your thirst neurotransmitter. So anytime that you're urinating a lot, or a lot of times if you're hemorrhaging, or if you have different types of diabetes, the angiotensin a lot of times will help trigger you to drink something to help kind of maintain that balance. Cholecystokinin, this is your satiation hormone. This is the one that tells you to stop eating. This signal basically gives you a feeling of feeling full. Neuropeptide Y, this is one that helps you feel the hunger pains. Also plays a role in the stress response too. For some people, the neuropeptide Y goes in overdrive, and this is why some people go into what we call stressful eating. Let's talk about neural circuits. There's 4 types of neural circuits, and this is going to be a very simplified version of what we see in and around the brain. But we basically see 4 types. We see diverging, which means one neuron may synapse with multiple neurons or multiple neuromuscular junctions. We see this with the majority of most of your nerves that travel down the periphery of the upper and lower limbs. You have converging neurons. These are pathway neurons to a motor neuron, where they converge into multiple neurons that converge into a single neuron. You have reverberating neurons. These are going to be like a cyclical type thing, very important for breathing, short-term memory, helps control how well you wake up. Naturally, if there's any kind of injuries to these things, then you can have a lot of problems trying to catch your breath or have problems actually physically falling asleep or waking. And the last one, special one we're going to talk about are the parallel after discharge. These are only used for mathematical calculations, and here's what they look like. So first, letter A, you see a diverging circuit, so you see a single neuron that's going to cause a cascade to multiple neurons and then to multiple neuromuscular junctions. Letter B is just the opposite. You see converging, will come in. Some of these may be inhibitory, some may be excitatory to the output neuron. C, you have the reverberating circuit. You can see how this one kind of feeds back on itself to reestablish a memory or help to regulate different kind of processes, in this case breathing. And of course, letter D is going to be called the parallel after discharge circuit. These are very important for mathematical calculations. So as far as regeneration and repair, we touched upon this earlier in this particular chapter. As we grow, naturally, your brain and your nerve system has a good amount of plasticity. As you're rapidly forming synapses, you're forming more brain-body connections, sprouting new dendrites, you have new proteins that are being synthesized constantly. You're creating neural pathways, just very simply from recognizing what you see to learn how to pick your head up, to walking, to later on learning how to ride a bike, a skateboard, to playing a sport, to balancing, a whole slew of things that happen through here. Unfortunately, once we hit a certain point, that plasticity starts to drop, and then we have a very limited ability to repair and or regenerate certain areas. So again, in the peripheral nervous system, you can repair damaged dendrites and axons, but certain things have to be in place. Currently, in the central nervous system, there are no repairs are possible except in the hippocampus, which we'll find deep in the limbic system of the brain. We'll talk about that in a different chapter. Surprisingly enough, that does have the capability of doing mitotic division, but so far that is the only area of the brain that has that capability so far. So repair within the peripheral nervous system, we're going to see two different processes that are happening here. So number one, like I said before, the cell body has to be intact to regenerate the axon and or the dendrite. The Schwann cells have to be able to be present. They must be patent and form a tube called a regeneration tube, and scar tissue can't form too rapidly. So for some people, especially if you're dealing with, say, like a stroke victim, which we've had in the past, or someone has a car accident with nerve damage, it's important to get them started on rehab very, very quickly to basically alert the body, very similar to what we talked about with Wolff's Law, with laying down more pressure onto a bone to allow it to form, or Davis's Law, which is going to be the soft tissue version. You want to at least trigger the brain to realize we do need the structure, start laying down more tissue to reestablish that connection. But once happening is within about an hour, assuming a day or two after injury, the Nissl cell bodies will start to move towards the periphery. They'll start to break up the fine granular masses to basically pave the way, break down the necrotic tissue, and basically start to lay down more tissue down the regeneration tube or down the myelin tract. So usually within 3 to 5 days, you have Laurian degeneration occurs, which we see a retrograde degeneration. So what happens is if you injure one node at the end, it goes back one node. It basically will tear down the axon there, use the myelin sheath to start laying down more tissue there, and start growing the regeneration of the axon from the periphery. From the nerve cell body down the axon hillock towards the periphery, towards the terminal end bulbs. But it's slow. It generally goes roughly about 1.5 millimeters per day. I've seen it go as high as 5 millimeters depending on what kind of text you're reading. But you do need the Schwann cells there to undergo mitosis to keep laying down the track, so to speak, or tube, to allow proper regeneration. Okay, this is the last slide of the chapter. So we talk about neurogenesis in the central nervous system. Right now, the only neurogenesis that we see again is going to be in the hippocampus. When we're growing, when the brain is developing, we have what's called the epidermal growth factor, is constantly being secreted. But unfortunately, once we hit a certain point, to be between the ages of 18 and 24, that neurogenesis gets shut down. And scientists are not exactly sure why that's happening. We think it's inhibition of neuroglial cells, or simply the genes just simply turn off with the green circulating factors. It could also be because there's a lack of neurolemmas or possibility of rapid formation of scar tissue, which almost acts like a block or inhibits any more cell production, whether it be new neurons, whether it be new neuroglial cells to help nourish those neurons as well. All right, so this finishes up chapter 12. This will finish all the material for exam number 4. Everything triggered after this, anything covered after this will be towards the final, but we'll use some of these terms for exam number 5. We'll talk about different areas of the central nervous system, peripheral nervous system, but for the most part, you guys are all set. Good luck with exam 4, and we'll see you guys in chapter 13.